Okay, so today we are going to look at nucleic acids, and these nucleic acids are polymers of a specific sequence of monomeric unit called nucleotides, which are linked together through 3' 5' phosphodiester bond. So what does that mean? It means that the nucleic acid is a large molecule, it's a polymer. When you said compound is polymer, it means that it's made up of smaller units, and those smaller units are called monomers. Like for example, in the case of DNA, as I said, we have monomer, we have monomer. So a monomer produce what you call polymer. It produce what you call polymer. So monomer produce polymer. This is the general concept from chemistry. It's a general concept from chemistry. We have monomers, and these monomers produce polymer. And although we have different types of reactions that involve the process of the formation of this polymer, we have what you call polymerization reaction. We have polymerization reaction, and apart from polymerization reaction, we also have condensation reaction. We have condensation reaction. So what is the difference between polymerization and condensation reaction? The difference between polymerization and condensation reaction is that in polymerization, we have, in polymerization, it's just that monomers are produced, are joined together to produce a larger molecules without the release of any small molecules. But in the case of condensation reaction, it's also like a polymerization reaction, but the only difference is that a larger molecules are produced with the release of smaller molecules. It means that each time a monomers joined together, small molecules are released. So that is condensation reaction. So as I said, we have different types of polymers. We have different types of polymers. Like in biochemistry, we usually talk about four basic polymers, or let me just say three basic polymers. We talk about these polymers. We have carbohydrate, CHO, carbohydrate, we have proteins, and then we have the nucleic acids. We have proteins and then nucleic acid. So in biochemistry, we mostly talk about this type of polymers. We have nucleic acids, carbohydrate, and proteins. So in the case of nucleic acids, the monomers are the nucleotides. The monomers are nucleotide. It means that they are the smaller blocks that are used in building the nucleic acids. So we have polymers. So in this case, the polymers for nucleic acids, we have nucleotides. But these nucleotides are linked by a bond, and this bond that linked one nucleotide with another is called phosphodiester bond. So it means that if you have a monomer, let's say this is your monomer, and this monomer joined together. So as this monomer, which is, let's say this one is monomer 1 and this one is monomer 2, so they joined through these types of bond, which is the phosphodiester bond. And so also, at the same time, when these monomers are joined together, small molecules are released, and these small molecules is water. So whenever one nucleotide joined with another nucleotide using the phosphodiester bond, then a small molecules is released. And this small molecules is water. And therefore, we are not going to consider it as polymerization reaction, rather is condensation reaction, because smaller molecules are released during the course of the reaction. Okay, and then apart from this polymerization reaction, then we also need to understand something. In the in this nucleic acids, we have all they travel in different direction. They are in different direction. So we have 3' direction and 5' direction. So this is the 5' direction, that is the left, and the right is the 3' direction. So the direction of the phosphodiester bond is in 3' to 5' direction, and that is why it's called 3', 5' phosphodiester bond. So I think from this slide, you will be able to understand like the following concept. Number one, the nucleic acid are made of nucleotides, and the nucleotides are the monomeric unit, which is also considered as the monomers. And then we have the polymers, which is the nucleic acids. And these monomers are linked together through phosphodiester bonds in the direction of 3' to 5'. And most of the monomers we talk in biochemistry, most of the polymers we talked in biochemistry is carbohydrates, proteins, and nucleotides. And these monomers are usually joined through a condensation reaction. And a condensation reaction is a type of reaction in which small molecules is released when the monomers are joined. Okay? So now, what are the components of this nucleic acid? What are the components of this nucleic acid? The nucleic acid, as we said, is made of nucleotides. But before you have a nucleotide, you have a nucleoside. So now we are going to look at the difference between nucleosides and nucleotides. So nucleoside is a combination or is made of two components. And this component of nucleoside is sugar and base. But in the case of nucleotides, apart from the sugar and the base, there is also a phosphate group. So it means that the difference between nucleotides and nucleoside is just the presence of phosphate group. And now, if you can now look at this, so this is a nucleotide. This is a nucleotide. So you see, as we said, nucleic acid is a polymer of nucleotide. So you see here, if you can clearly see, is one nucleotide because this is a nucleotide. All this here is nucleotide. So we have one nucleotide here joined with another nucleotide here, then another nucleotide here, then it's now more are added till you get the nucleic acid. So ladies and gentlemen, in this point, you should understand that we have nucleosides and nucleotides. And the nucleoside is made up of sugar, base. And then for the nucleotides, we have sugar, base, and phosphate. And for the nucleic acid is made up of repeating units of these nucleotides. Okay. Okay, now we have two types of nucleic acids. We have two types of nucleic acid. And these two types of nucleic acid, we have DNA. And the DNA stands for deoxyribonucleic acid, while RNA stands for ribonucleic acid. So these are the two types of nucleic acids that we have. And these nucleic acids, like for example, we have it started from the DNA. And if you have a molecule of DNA, the process in which one molecule of DNA copied itself to produce another molecule of DNA, the process is called replication. So if DNA copy itself to produce another molecule of DNA, then the process is called replication. So the conversion of or the copy of one DNA to another molecule of DNA is called replication. But when this DNA molecule is converted to produce RNA, then the process is called transcription. So the copy of mRNA or the copy of RNA from DNA is called transcription. And then the copy of RNA to protein is called translation. So all this together, we call it central dogma of molecular biology. All this is called central dogma of molecular biology. We call it central dogma. We call it central dogma of molecular biology. So the central dogma of molecular biology comprises of replication, molecular biology. So we call it central dogma of molecular biology. So the central dogma of molecular biology comprise of replication, then two, transcription, and the three, translation. So these are the central dogma of molecular biology. Okay, and then these nucleic acid are serve as a repositories and the transmitters of the genetics information. So what does that mean? It means that all the genetic information that the child inherits from their parent are resides in these nucleic acids. Like some of our child, they look exactly the way we are, in terms of color of skin, in terms of height, the way we talk, in terms of intelligence, in terms of many physiological characteristics, as well as even anatomical features. So all these hereditary information or all this genetic information are there in the DNA. So DNA is what make our child look like us, and the DNA is what make one organism differ from one another. If you are smart, it's because of your DNA. If you are intelligent, it's because of your DNA. If you are if you are if you are if you have fair skin color, it's because of your DNA. And if your skin color is black, it's actually as a result of your DNA. If you talk too much, it's actually as a result of this nucleic acid material. If you are very sexually active, it's because of your DNA. If you are dull, it's because of your DNA. And that is why it is always advisable, even if you are going to even if you are going to marry, it's better you always try to have a different combination so that at least your child will come up with a very beautiful characters, as well as the morphological, or let me just say genotypic and phenotypic characters. Because when we have a child, so a child is a product of the genetics recombination, or it's a product of combination of our own DNA. So the DNA from the father is coming, and then the DNA from the mother are also coming to fuse themselves together to produce another DNA, which will actually produce your own child. So DNA is very, very important. It's very, very important. In fact, the characters of stealing is also there in the DNA. So, ladies and gentlemen, DNA is very, very important. So, it is the repositories and the transmitters of genetics information. That is why last time I said that DNA is actually the language of life. You look the way you are because of your DNA. If you are smart, it's because of your DNA. And if you are not smart, it's because of your DNA. In fact, even in the case of drugs that we are taking different types of drugs. Some drugs may be active in some particular individual. While when this different individual take the same drugs, it may have different action. And this is all reside in the DNA, because all the characters and the habits are there are as a result of the proteins that are actually expressed or produced from that particular cell. Hemoglobin is responsible for the transport of oxygen because of the presence of the hemoglobin, and hemoglobin is a protein. The brain is responsible for the controlling of actually the memory as well as the intelligence, and is all as a result of the proteins, and all those proteins are produced from the DNA. So some proteins are produced in higher concentration, that is in higher quantity, compared to those actually other organisms, or sorry, other humans. Like for example, someone, or you can have a particular individual where the concentration or the amount of the neurotransmitters as well as the proteins in the brain produced are very low, while in some other individuals you see that proteins and the
Mm-hmm.
neurotransmitters are produced in larger quantity. So as a result of that, you can't expect the intelligence to be the same. So DNA is very, very important, ladies and gentlemen. Okay. And then now, what are the functions of this nucleic acids? So number one, DNA is the chemical basis of heredity. That is what I just explained. So it's a chemical basis of heredity. It's responsible for controlling heredity. So if our child look like us, it's because of the DNA. But whenever there is a problem or mutation in the DNA, that is where we have variation. That is where our child will look different from the way we are. And that is actually as a result of the changes, as a result of some changes that actually happened in the DNA. And that is why sometimes, and also at the same time, when we may have some characters, because uh because the our child is actually a combined form of our own DNA. So when DNA of the mother and the DNA of the father joins together, they may probably produce some characters that may be different from either of the parent, okay? And then there are also reserve bank of genetics information. We also talk about this. They are the reserve bank of genetics information. So all the genetics information that we are talking about are reserved in the nucleic acid, and it's responsible for maintaining the identity of different species of organism over millions of years. Like now, for example, the first living organisms that exist on Earth, or the first living organism that exists on Earth is prokaryotic cell, which is bacteria. And it's about 3.9 billion years ago. Since that time, we still have prokaryotic bacteria, and they are all the same. The prokaryotic bacteria we have now and that of the 3.9 billion years ago are the same. And and that identity, that identity of the bacteria are actually maintained as a result of this nucleic acid. Like for example, although if we can check for the morphism, sometimes we may actually experience some changes. Like the first human species that exists on Earth is about, I think, around 400,000 years ago according to evolution. So now you can actually look at the differences as well as the similarities. So that similarity between the first human that came around 400,000 years ago and the current humans that are surviving currently, or that are surviving now, you can see that the difference is very small. And all those identity are retained as a result of this nucleic acid material. And then cellular function is under the control of DNA. So all the function of any cell you know in our body, we have like 200, we have like 220 different cell types, like brain cells, kidney cell, eye cells, skin cells, pancreas cells, and kidney cells. All of them, they are different cells, but they are controlled by DNA. And the kidney cell is different from the liver cell because of the control and the regulation of the DNA. Because in some cell, some proteins will be allowed to be produced, and in some other cells, some proteins will not be allowed to produce. So as a result of that differential proteins production in different cells, that is what make different cells differ from one another. Then the basic information pathway. The DNA is responsible for actually producing the basic information of a pathway. So actually at this point, you may not actually understand the pathway because pathway is we usually talk about pathway when we are talking about metabolism, but I think you can have this basic knowledge from secondary school. We have glycolysis. So glycolysis is a pathway. And then we also have gluconeogenesis. We also have citric acid cycle. All these are the pathways, but they are all controlled by the DNA. And the DNA direct the synthesis of RNA, which in turn direct protein synthesis. And this is what we discussed. From DNA, we get RNA. And from the RNA, we get protein. Okay? So now, we are now going to look at the basic nucleic acid structure. So remember that we said that the DNA is a polymer of nucleotides and it's held by 5' and 3' sugar phosphate bridges. So you see, as I explained, this is a nucleotide. This one is a nucleotide, and this one is also another nucleotide. This one is also another nucleotide. So that is how it moves. So these are the nucleotides. So they are all linked. They are all linked by 5' and 3'. Please, who is that making? So this is how the nucleic acid structures look like. So one nucleotide joined with another nucleotide is like the previous explanation. Okay? So now the composition of nucleic acid. So nucleic acid, as we said, is a polymer of nucleotides, and the nucleotide is made up of nucleoside and phosphates, and the nucleosides is made up of bases and sugar. And these bases, we have two types of bases. We should all know. We have two types of bases.
Can you
Number
say
one,
that
we
nucleosides
have,
cannot
please
be
let me finish.
mixed,
After
like
we finish
joined
the class, we
together?
ask question.
Something like that.
So basis is made up of furans and pyrimidines. And then for the sugar, we also have two types of sugar that are found in nucleic acid. We have ribose sugar and we have deoxyribose sugar. So the ribose sugar is found in RNA, in DNA, in RNA. The ribose sugar is only found in RNA. So we have ribose sugar in RNA. Please, we should put this at the back of our mind that the ribose sugar in is only found in RNA, while the deoxyribose sugar is found in DNA. So please, I think since from now you will begin to understand some basic difference from DNA and RNA. That the RNA is having ribose sugar, while the DNA is having deoxyribose sugar. So it means nucleotides is nucleoside plus phosphates. And then the nucleoside is bases and sugar. And the bases are purines and pyrimidines. And for the sugar, we have ribose sugar and deoxyribose sugar. And the difference between this is the ribose sugar is found in RNA and the deoxyribose sugar is found in DNA. Okay, then the nucleotides. So nucleotides, as we said, are the building blocks of nucleic acids, and a nucleotide consists of three fats. These three fats comprise of the heterocyclic bases, sugar, and phosphate. So you see, this is the pentose sugar. So the ribose sugar is a pentose sugar. It means it's a sugar with 5 carbon. And this is the nitrogenous base, and this is the phosphate group. So we should know that. Remember in chemistry, when we said heterocyclic base, when we said heterocyclic compound, it means that are compounds that have other atoms, other atoms in the ring, rather than also the other atoms apart from carbon and hydrogen. It means that they have other atoms apart from carbon and hydrogen in the ring, because we are talking about cyclic. So whenever we said cyclic, it means that we are talking about the compounds that are cyclic in nature. So it means that in the ring, it means that in the ring, they have other atoms apart from carbon and hydrogen. Mostly in this kind of bases, the other atoms that are found apart from the carbon and hydrogen is nitrogen. That is why they are called nitrogenous bases. That is why they are called nitrogenous bases, and we are going to look at them